Again, welcome to the GC University Hyderabad channel on YouTube. Dear students, we are continuing our lectures for the BS students of botany as well as major subject as well as for the minor subject. For the BS Botany, not BSc, but the BS Botany. Again, welcome. We have already discussed in vascular plant the diversity of the vascular plant, and we have discussed the psilopsids, lycopsids, phenopsids, and in vascular plants in my last lecture I have discussed the stellar system. Now we are coming to the fourth group of this vascular plant, that is Pteropsida. Pteropsida. Next, the Pteropsida, so many orders are here in this Pteropsida, but the familiar plants of this Pteropsida are ferns. Last time I have told you that we have divided this into the Pteropsida into Felicini, last, Gymnosperm, Angiosperm. The Pteropsida, Felicopsida, according to the Pitchy Ceremony 1959, Pteropsida classified into 7 classes. We will follow the Raman classes on Raman classification in 1954, which he described. The order Cynopteridales comprises only of the fossil members, and in the last Paleozoic era they were present on the earth, and they dominating on the earth in the late Paleozoic era. The families in which this order has been divided: Prof, Zycopteridaceae and Calidoxylaceae. The Zygoteridaceae, where the fern belong, and these are the living plants of the Pteropsida. That phylogeny means it belongs to the group. The ancestor of this group are the Xylophytes. If we go to these plants, these are highly evolved. They belong to the highly evolved vascular plants. The highly evolved vascular plants are Pteropsida, Gymnosperm, Angiosperm. Although it does not produce the seed, these plants of the Pteropsida do not produce the seed, but they belong to the highly evolved group of the vascular plant. They, as I have told you, the most beautiful plants are the ferns. You will find, or I have seen these plants, especially in the hilly areas. Under the pinus trees, you can see lush green ferns on the ground in the Azad Kashmir Valley. You can go to the Nathia Gully and somewhere else, you will find under the pines, and the pines means the Conifera. Under these Conifera, these plants are growing thickly, luxuriantly. Especially these plants are considered as the hygroscopic plants, meaning the moisture-loving plants. The living ferns have nowadays 305 genera, and near about 10,000 species of these plants are present. Nowadays, for long since Carboniferous period, we have discussed the Phenopsida last plant, which were luxuriantly grow and dominating earth in the Carboniferous period, but this evolved from that period, distinguished from the Lycopsida and Phenopsida in different vegetative characters. First vegetative character which is distinguished here, that it is the start of the megaphylls, means multi-veined leaf, or we can say the large size leaf like that. These are the multi-veined leaves. You can see the leaves here, and so many veins are present, but before that only and only one vein was present, but here we have number of the vein and veinlets present in this. You can say or see also these clearly here. So this is megaphylls. And another type of the megaphylls are here. This is single leaf, you can see, which is divided. Its lamina is divided. This is the lamina of the leaf, which is divided, which is in one piece. That's why it is one piece, and that's why it is called the simple leaf. But here this lamina, the same lamina, is divided into number of the leaflets. That's why we can say that it is not a simple leaf, it's a compound leaf. But you can also see number of the veins and veinlets into it. So both belongs to the megaphylls. Now after that, you have seen the difference between the leaves of the Lycopsid, Phenopsids and Pteropsid. The Pteropsid is the beginning of the megaphylls, and you have seen the different type of the megaphylls with the number of the veins. Alternate leaf gaps are also present here. If you have studied, last time we have studied the leaf gaps in our stellar system. So the alternative leaf gaps are also notable in the anatomy of their leaves. So reproductive features, if we go to the reproductive features, here the sporangia are aggregated in a specialized type of the structure, and these structures are also present at the foliage leaves. Last time we have studied especially the Lycopsida, Psilopsida, Phenopsida. There are a number of the brown leaves which form strobilus, but here the strobilus are not formed. These special structures are formed, different type of these structures are formed which have the aggregation of the sporangia, and this aggregation of sporangia which are present and specialized structure present on the foliage leaves. So we can say that special strobilus or any type of these structures are not present for the sporophyte. And we also know that the main plants of all vascular plants are the sporophytes. So they have different type of the structure, vegetative structure, stem, root, leaves, but they have a specialized structure for their reproduction, which is called the sporangia, and they produce spores for the asexual reproduction. So here the aggregation of the sporangia on the abaxal or maybe adaxal surface. Abaxal surface yani at the axis or at the base of the leaf or somewhere at the lower side of the leaf. Next is the stem. If you will see in ferns, we have different type of the stems, but most of them are underground rhizome structures, means they move horizontally in the ground. We can see the rhizome here. We can see as well as rhizome here, but with these rhizomes, the hairs-like structures are also present. Sporophyte has underground rhizomous stem, may be elongated or tuberous, means somewhere the tuberous structure, you can see here, the tuberous structure are present, means they aggregated and form a tuber-like structure. Branching, if you will see here the branching, you can see this is the leaves are directly attached with that, and if you will see here the branching are alternate, so dichotomous or alternate. In some cases rhizome is covered by hairs, as I have told you before that the rhizome are covered by the hair-like structures. Now after that, if we see the leaf, we have discussed just now that the leaves are metaphylls. They are compound leaves. I have told you that it is simple leaf. This is simple leaf. That the leaf has one single lamina. The lamina is not divided into number of the leaflets, but number of, here if you will see, it is one leaf. Don't consider it number of the leaves. It is single leaf. The stipule is present at the base of it. This is the apex of the leaf. But the lamina is divided into number of the leaflets. It's a single leaf. But one, it is divided into just one part and one part. So this is called the unipinnate compound. If these pinna divided further into number of the pinnas, then they will be called bipinnate leaf. So the compound leaf may be unipinnate or may be bipinnate. If you will see here, we can see the unipinnate leaf. If you will see here, we can see the bipinnate leaf. So two types of the leaves are present here. The compound leaves, either unipinnate or bipinnate megaphils, whose reticulate venation size varies. You can see the size of that, a large size, or you can also see the small size here. So the leaves are aerial whenever the stem is underground, present inside the ground, but leaves are present in air. So the things which are present in air are called aerial. Rest of the plants are subterranean, means ground, on the ground. Some forms show succinate. Succinate means they are present in the form of the circle. This is called succinate. I will show you later on what is succinate venation. Whenever it will open like that. Or maybe on the other hand side, it may form a circular structure whenever it is in a bud form, but whenever it will open like that. I will show you later on. Some forms show succinate venation, coiled inward in young, and uncoiled means inward coil, and whenever it will open, it will outward. So that is called inward in young and outward in the older one. So this is the structure which is written here. Now you can say that this is succinate, as I have told you. This is succinate venation. This is succinate venation. This is succinate venation of the leaflets as well as the leaf. Now after that, as we have studied that all psilopsids, lacopsids, phenopsids, pteropsids don't have seeds. If the seeds are not present, the taproot never develop. So the plants which I have given you the name, the groups of the plant, they do not produce the taproot, they always produce the adventitious roots from the stem, from the rhizome, from the stem they have the structure which are called the adventitious roots. Wagner, the trophopods. What are trophopods? Wagner and Johnson in 1983 reported a special food storing organ trophopods in number of the form such as asprillum or platineuron, onocelia, and Dryopteris. You can see here, you can see here some of the food storing organs, and these food storing organs are called trophopods. It is reported by the Wagner and Johnson. Next after that, if you will see the different plants of and their leaves, I have given a pause for the Azan because Azan was coming. So now we are again continuing our lecture after a little break. These are the different Pteropsids plants, Dryopteris, which you have already studied in your first year. The onocelia, another type, you can see the differences in their leaf structure. Another platineuron and asplenium trichomonas. Now, after that, now there is one. We have already discussed the stellar organization in my last lecture: protostine, solanostine, polycyclic steel, dictyostene. These different type of these steels are present. Their rhizome is stem, because the stem is type of the cortex. If you will see here, in especially Ophioglossum, these are the plants of the Ophioglossum. If you will see here, the cortex, there is something wrong, cortex, cortex, the parenchymatous tissues are present in this one, and then they are maybe distinguished into outer parenchymatous zone as well as the inner parenchymatous zones. So outer side, somewhere the parenchymatous tissues are present outside, and somewhere the parenchymatous tissues are present. Here you can see simply that stem are present inside the ground, and the leaves you can see very simply the wing leaves are present here. Now, some tissues, mucilaginous tissues are also present. Sometimes mucilaginous tissues are also found, especially in Angiopteris, and then xylem as tracheates. Generally in Psilopsids, Lycopsids, Phenopsids, Pteropsids, the xylem consists of three elements: tracheates, fibers, and parenchymatous tissues. While in angiospermic plants, the vessels are also present as a xylem element, but here in some plants we can also observe the vessel in their xylem. They are also reported in Marziella, especially one of the fern plants. The secondary growth generally absent because the stem is rhizome type, so the wood cannot be formed properly. But here, especially in Botricums, you can see, this is the Marziella plant, but in Botricum we can see the secondary growth up to some extent in these plants. Then after that you can see the structure of the leaves and the structure you can see here. This is the Botricum plant, and you can see the branches of that, and here it is shrub-like a structure. That's why you can see the secondary growth here. That is the Angiopteris. In Angiopteris the leaves are different, and you can see the spore-producing structure at the border of it. In some plants you can see somewhere these spore-producing structures. Roots. If we go to the internal structure of the root, still is usually protostelic, the root, in root, with the variation in xylem. You can see the exarch xylem are here. These are the exarch, means the phloem is outside and the xylem is inside. But we can also see the mono, tri or even tetrarch. You can see this is tetrarch, and the stele is protostele. There is no pith in between it. These are the xylem and form four structures here which are joining in the center. The phloem is alternate with each other. You know that in the roots the radial vascular bundles are present, and here you can see clearly these are the phloem and here the xylem. But the xylem form, if it will form two structure, we call it the diarch. It forms the three structures like that, we call it triarch. If it is single one, we call it the monarch. So these are the different types which can present in the roots of the Pteropsida. Then after that, we can see the reproduction. You know that in all plants, the reproduction of plant is an alternation of generation. In sexual reproduction, alternate with the sexual reproduction, and I have discussed in all of these plants of the vascular plants that the main plants are sporophytes, so it performs asexual reproduction. So many ways of asexual reproduction are present in plants, but the usual type or the routine type of the asexual reproduction occur in plants during its life cycle is the spore formation. And why spores? So every plant or every sporophyte has a specialized structure which is called a sporangia, and this sporangia produce spores by meiosis. I have also discussed in my previous lectures that the meiosis always takes place once when the plant produces spores, and the spore mother cell is responsible to produce spores. Sometimes these spores are morphologically similar, sometimes these spores are morphologically dissimilar. If the plant produces morphologically similar spores, they are called homosporous plants. If the plant produces morphologically different type of the micro and megaspores, then the plants are called the heterosporous plant. These are homosporous plants, and they produce the spores, especially as usual in sporangia. But I have told you earlier, the sporangia are aggregated in a structure which have different names in different plants. So the asexual vegetative reproduction, you can cut a plant, put into soil, plant part, vegetative part, put in the soil, and it will fragment by the process of the fragmentation, this vegetative propagation method. You can also say fragmentation. Adventitious buds, you can take the adventitious roots in the form if they are present in the buds. The embryonic leaf apices, some of the leaf apices in the beginning may work for the vegetative part of the propagation. Stem tubers, root tubers, these all is structured, but I have told you the spores are the usual way of the asexual reproduction in plants. Here, now we are going to end this part of the Pteropsida, but still the Pteropsida continue in my next lecture. So, if you have any queries, you may ask to me by SMS or by WhatsApp or message on the YouTube as well as. Thank you very much.